Okay, it's time for our next session. So this is session number 4, experimental modeling and genetic manipulation in human brain slices. And yeah, we have 2 speakers for this sessions, and we already have learned in talks from Jonathan, for example, that you can modulate gene expression, specifically in cell types that you're interested in, and then will also lead to functional changes. This is very exciting. And so our first speaker will be Dr. Sam Brooker from the University of Edinburgh, and he will talk about genetic manipulation in human brain slice culture, and I think he also will make a nice announcement.
Maybe. Maybe. Good afternoon. I'm just going to make sure this is working for me. There we go. Easy. So hi. It's a pleasure to speak to you today. I was meant to be Claire Durrant. Unfortunately, she couldn't make it, so I'm going to present some work that we have, our respective lab groups have kind of generated together. So this is the Edinburgh Human Brain Group at our annual meeting a couple of months ago. And I'm Sam, and I'm from the University of Edinburgh, and I work largely in the Simons Initiative for the Developing Brain. And I'm going to talk to you about our efforts to model genetic neurodevelopmental conditions in adult human brain tissue. So before I get started, I really want to attribute the effort that's gone into this. This work's been largely led by an extremely talented PhD student in the lab, Max Knops, with support from Soraya Mefta, who has recently been awarded funding to start their own lab. And this work was all bioarchived last year, so if you're interested, I recommend going and having a look, and we're currently under review, in revision, so hopefully the final manuscript will be out in the next little while. So why neurodevelopmental conditions? So these affect about 2 to 4% of the global population, and from lots of incredible work from the Simons Foundation Autism Research Initiative, we know that there's a genetic basis. And based on this, on the Safari gene list, we know there's about 1,400 genes that are associated with neurodevelopmental conditions and neurodevelopmental delay. And these genes have roles both in neurodevelopment, but also neuromaintenance as well. So how the brain develops, but then also how the brain maintains itself throughout life. And we know from lots of work, particularly from rodent studies, that loss of function of these genes, or deletion of these genes, or alteration of their expression leads to changes in cellular excitability. And this change in cellular excitability may be one of the key phenotypes that's giving rise to the many of the experiences that the individuals with these conditions have throughout their day-to-day lives. So I'm going to talk to you today about one condition in particular that I've worked on since I joined Edinburgh 12 years or so ago, which is Fragile X syndrome. So Fragile X syndrome affects about 1 in 6,000 people around the world. It's X-linked due to this fragile site on the X chromosome, and as a result, it affects male births slightly more, about twice the incidence as female births, so about 1 in 4,000 live male births versus 1 in 8,000 females. It results from loss of function of the FMR1 gene during embryonic development. And this loss of FMR1 leads to loss of the protein FMRP, which leads to intellectual disability and sensory processing difficulties in most individuals, as well as comorbidities with autism and epilepsy in about 30% of individuals, respectively. And we know, like I said, we know a lot about the function of FMRP from rodent studies. So from a really great review from Vitaly Klyachko a few years ago now, highlighted that we know that FMRP's canonical function has been attributed to altered translation of proteins, as it's a ribosomal repressor. It's also associated with protein-protein interactions, most noticeably through potassium and sodium channels, as well as altered trafficking of proteins and altered phosphorylation. And a few years ago, we tried to summarize the key cellular structures that display altered function from these rodent models. And I think it's pretty clear to see that right from the synapse through to the spine, dendritic branch, soma, and axonal segment, as well as the distal axon, there are significant alterations in function. But I don't need to explain to this audience that many of these structures display species divergence with the human brain. So if we want to understand what's happening in Fragile X syndrome, we really need to be able to understand what's happening in these different cellular compartments in human neurons. And as I alluded to, Fragile X has been modeled across the evolutionary spectrum. So there are C. elegans models, fruit fly models, zebrafish models, particularly mouse models are very predominant, and increasingly so rats, as well as there are marmoset models. And there's been an increased use of patient-derived iPSCs and in two-dimensional cultures as well as organoids, which have allowed us to get great insight into the biochemistry of FMRP. However, all of these models lack the cellular diversity and function that's present within the human brain. So, and this, we believe, has led to the translational bottleneck in Fragile X research. So, since the FMR1 mouse was developed in the early 90s, there have been many, many clinical trials, pretty much all of which have failed. And this highlights a greater need to link lab research to the clinic and understand the role of FMRP in human brain circuits in particular, both in development, but also beyond development, given the implications of re-expressing FMRP, particularly in gene therapies, or the effect of aging as we have an increasing aging population. So what approach have we taken? So about 7 or 8 years ago, 5 or 6 years ago, when Claire Durrant joined Edinburgh, we started, her lab started generating brain slice cultures from glioblastoma and glioma patients, primarily from the access tissue overlying these deep-seated tumors. We then slice them and culture them using a modified brain fizz-based approach. These cultures are then plated. We have matched paired cultures from each patient. We treat some with a scrambled GFP construct and some with an FMR1 shRNA GFP construct. These are all short hairpin RNAs under the U6 promoter, so it targets everything, mimicking the case of Fragile X syndrome, where FMR1 and FMRP are ubiquitously expressed. We want to knock it out in everything. And we're using lentiviral vectors here so we can transduce the cells in as minimally invasive and toxic way as possible. We allow these cultures to age for up to kind of 8 to 10 days, and these were treated either with 1 micromolar or 1,000 micromolar dibutyl cyclic AMP. And I'll come into why that's important in a minute. So first of all, we wanted to validate this model. So we started off, we did some immunohistochemistry. Here's our GFP labeling from our viral vector. Here's the endogenous FMRP in a labeled cell, and this was in layer 2-3. And you can hopefully see this pretty well aligns with the cell body and dendritic structures. When we look at our knockdown cells, hopefully it's reasonably clear to most people that there's much less of this pink FMRP stain co-localized with a GFP positive cell. So we quantified this, we max quantified this, and so what you can see here in the black line are the scrambled cells. These are our controls. We have the GFP negative cells, so these are just presumably wild-type human cells, and then we have the knockdown cells here in red, and you can see there's a leftward shift indicating lower fluorescence intensity at the cell body. And then we quantified across all patients we tested with this. So we have 9 patients for the scrambled and the shRNA, and we can see, hopefully it's clear to see, we see a pretty robust knockdown after 7 to 8 days in vitro. Well, 8 to 10 days in vitro, sorry. So this is cool. We can knock down FMRP. We can generate a situation where certain neurons in our slice cultures have lower FMRP, which might mimic aspects of Fragile X syndrome. So next, from a slice physiology perspective, we wanted to ask what's the underlying physiology of these neurons. So Max performed whole cell patch clamp recordings from neurons in layer 2-3. I can go into it later, but we had a lot of exclusion criteria to try and really make sure we were targeting neurons, not astrocytes or transforming glioma cells or anything else that could be in the tissue. So we had a lot of exclusion criteria, and hopefully it's clear we can get nice neurons that fire transvaction potentials. And when we looked at the FMRP lacking neurons, we found that these tended to fire more action potentials than our control cells. And across the range of cells we recorded, we see that's pretty robust and consistent. And what's really nice about this is this mimics pretty well what we see, what has been reported in rodents. So what we think is we have a good model where we can see a parallel hyperexcitability at the single cell level, but in a human model, which allows us the ability to then start to think about, can we understand what FMRP is doing in the human brain? So first of all, we want to ask, well, what's happening at the level of the action potential? Full disclosure, the action potentials in our cells are kind of small and slow. We don't really know the reason why that's the case. But when we look at the FMR1 knockout cells, by knockdown cells relative to the controls, which are the reds, knockdown, blacks, the controls, we can see the knockdown cells are larger and they are faster. And this is reflected in the phase plot here. And when we quantify that across all of the cultures we've recorded, cells in all the cultures we've recorded, and these are linear mixed effects model stats here, so we've accounted for patient and matched within case, we can see that the RIA base is reduced in these cells, voltage threshold, although not significant, is tending down, maximum rise rate's much higher, and maximum decay rate's also much higher here. Interestingly, we wanted to then ask, well, what's happening across our patient demographics? We've sampled from lots of different patients. Is it consistent? We know it's X-linked, but is losing FMRP affecting males and females equivalently? So here we have our scrambled cells, and we only have a few females here, unfortunately, but we can see that under control conditions, male and females tend to respond with a similar number of action potentials, and this is uniformly higher in males and females lacking FMRP. And then we wanted to ask, well, is this affected by seizure phenotype? And again, we see a maintained effect of FMRP loss, irrespective of whether the patients have had experienced seizures preoperatively or not. And importantly, we want to ask as well, we know that FMRP has different functions in different brain areas depending on cell type. So do we see different effects in different brain areas? So we looked across the 4 brain areas we regularly get tissue from. So we have temporal, occipital, parietal, and frontal. And we see, by and large, the same pattern of increased activity is present irrespective of brain area. However, it's important to note that we see in our cultures big variance in terms of the firing output of neurons depending on brain areas sampled. So beyond excitability, FMRP has been implicated in changes in synaptic function. So for all of the cells Max recorded, he generated a brief spontaneous EPSC recording to kind of capture the synaptic diversity. And he found examples, reasonably nice examples of spontaneous EPSCs in both culture conditions. And when he quantified these, somewhat surprisingly, we actually found no difference in the synaptic properties of these spontaneous EPSCs. So the amplitudes were much the same, and the frequency was much the same. So who knows what's going on there, but I think there's a lot more we can do in this space to try and understand how synaptic function, in particular plasticity, is changing. This is something Max is chasing up at the moment. But what was interesting for us is we now have a model where we can start to think about testing therapies and therapeutic ideas that have been put out in the Fragile X field for a number of years. And one of the routes we wanted to start with, largely based on my current funding, but also kind of ongoing clinical trials, is that it's been known very early on from patients with Fragile X syndrome that they have altered levels of cyclic AMP when measured in the blood as a biomarker. And so a recent clinical trial from Liz Berry-Kravis at Rush has shown that there's potential promise of increasing endogenous cyclic AMP levels through inhibiting endogenous phosphodiesterase pathways. So we wanted to ask, well, what happens if we increase cyclic AMP? Do we see some recovery of this excitability that might be parallel for altered circuit function? So what we did was, first of all, we want to ask, well, what happens if we lower cyclic AMP in the first place? So we took our normal brain fizz, which has got 1 millimolar dB cyclic AMP, and we reduced that concentration down to 1 micromolar to know whether or not there's a dynamic effect of cyclic AMP. And rather satisfyingly, we actually still, even though we have lower concentrations of cyclic AMP, we still see this hyperexcitability. And this is underlain by the same biophysical properties we had earlier. And then we next asked, well, okay, this effect of excitability is independent of cyclic AMP signaling, seemingly, but if we increase cyclic AMP in the knockout cells, can we correct function? And that seems to be the case. So here we've applied the same phosphodiesterase inhibitor that's currently under clinical trials, and we then measure the same excitability output. And whilst we see some effect in the wild type or the control cells, which is not unexpected, this is biologically active, we see a strong reduction in the action potential output of these knockdown cells. And finally, what's really, I think, really interesting, and this only came out, we only got this data about a week ago, when we do the same BPN phosphodiesterase inhibition experiment in these lower cyclic AMP cultures, we don't see this rescue, which to us is indicating that potentially these cells, we need to have a high concentration of cyclic AMP to reveal the underlying pharmacological mechanism of action of this drug. And I'm going to skip over that. This is the last little bit of data for this. Sorry, I won't skip over it. The last question, this was reviewer 3's question actually, was what could be the mechanism of this? So we asked, cyclic AMP activates PKA, we know this. We know that PKA phosphorylates sodium-potassium channels which contribute to the action potential. So if we block PKA, can we prevent this excitability as well? And that seems, blocking PKA did not seem to have an effect on the excitability in the knockdown cells. And I haven't shown the data here, but when we combined phosphodiesterase inhibition with PKA inhibition, we still see no knockdown of we still see no rescue of the excitability. So I hope in this brief summary, I have shown you that viral knockdown of genes are a tractable way of understanding neurodevelopmental conditions in human brain tissue. And actually, really importantly, there was a recent preprint that Jonathan highlighted earlier that was released this year that actually validates many of the findings we've observed. So actually, we're really quite confident that what we're seeing is biologically relevant. That study looked at, using a very similar method to us, looked at layer 5 of the cortex. So this might be cell invariant. Hopefully I've shown you that neurons lacking FMRP display increased excitability consistent with some of the rodent models, but I haven't had time to go into it today. There are differences between the rodent models as well. Lowering cyclic AMP still seems to maintain this biological effect, but the mechanisms of this are not entirely clear. And ultimately, blocking cyclic AMP metabolism might correct some of this, might be contributing to some of this hyperexcitability effects, possibly through pKa-dependent mechanisms. And I think it's really important that this highlights the utility of human brain slice cultures to identify disease-modifying pathways, test discovery science, and actually test some of the theories and hypotheses that have been developed from excellent work in rodents, but actually translationally validate these in humans so we can improve that transition from lab to clinic and ultimately the lives of individuals, and hopefully identify new and exciting disease processes that allow us better therapeutic strategies in the future. So there are lots of limitations, and I'm not naive to this. Obviously, this is adult tissue. We're trying to study neurodevelopment. What's happening in the brain early in development might be completely different. We don't know whether all the effects that have been observed in rodents are just compensatory. And we've got some strategies we're working on to look at that. We're missing key brain structures. We don't get access in Edinburgh to hippocampus or deep subcortical areas. And there's been lots of data from lots of people, including some people in the room, showing that subcortical structures, including striatum, might have alterations in function. And we haven't, in this current study, taken into account cellular diversity. We know that excitatory and inhibitory neurons have very different profiles in Fragile X syndrome, and this is something we're actively following up now. And what we're doing at the moment is one of the things we need to try and do is we are hoping to do is directly measure cyclic AMP using fluorescent tags. This is in collaboration with a group in Copenhagen, trying to find neuronal specificity. So we've just got a new virus using a human synapsin promoter to really target neurons only. And we're trying to correlate these with patient features to really identify, do individuals express different FMRP functions? So I want to end on the last couple of slides with a couple of bits of information. Claire asked me to present this, so the trade-off was I could talk today, but I had to give some of what she wanted to say as well. So fortunately in Edinburgh, we were just awarded funding to establish the Edinburgh Human Brain Cluster. So this is with Claire Durrant, who is meant to be here today, as well as our consultant neurosurgeon, Paul Brennan. And what we're hoping to do over the next 4 to 5 years is utilizing these models such as neurodevelopmental conditions and models of dementia and cancer, try and incorporate into it what we've now learned about the diversity of cells. So taking our resected samples, performing patient-specific transcriptomic fingerprints so we can identify, do individual patients have individual profiles of transcriptomes, and how does this correlate to the physiology, cellular and synaptic function, and then how does these changes inform the effects we observe in our slice culture models of human disease, really to be able to pinpoint where the variability in these models comes from. In addition to this, we've just in the process of establishing a UKRI human disease network in the UK. So this is hopefully beginning in July of this year. And the aim of this is to really bridge across, and not just the brain, but across multi-organ systems. As neuroscientists, we tend to forget about the rest of the body from the neck down. And actually what we're trying to do is integrate a multi-organ approach to improve the methods we use and to guide future disease models. And this is based on 5 groups at the moment in Edinburgh, Hull, Nottingham, and Leicester and Oxford. And hopefully what this is going to be starting to implement is new systems and technology development, leveraging the experience from multidisciplinary research routes. And this is hopefully going to lead to workshops, meetings, and outreach that I don't want to be too glib about it. I'm going to hopefully lean on many of you heavily to contribute to some of these workshops in the future, if that's amenable. And finally, we were discussing last night at the social about the possibility of where we hold the next of these meetings. And I know some suggestions have been floated as well. However, Edinburgh, I think, is a really nice option in a couple of years' time. So unless there are other options available, maybe we propose that in 2 years' time, if everyone's in agreement, we come to Edinburgh. Yeah, so let's meet in spring 2028. I can't guarantee we'll have no rain, but we'll have whiskey, so it's fine. And bagpipes, maybe. Yeah, finally, I really just want to obviously acknowledge people who've been involved in this. First and foremost, I want to acknowledge the participating patients. We can't do anything without their consent and the agreement, the implicit agreement to use their tissue for the research that we think is important. We have a really great Emerge research nurse team led by Emerge, as well as Max and Soraya, who have led on lots of this work. Our funders, and if you're interested, we have the bioarchive here. Hopefully you'll see it in print soon. Thank you very much.
Hey Sam, nice. Just thinking about the synaptic thing, and that is a prevalent thing in the mouse models or other models, right? But I wonder if this is to do with the age of the tissue. Has anyone done a mouse model which was induced post-development, and would your system be more comparable to that?
So it's very tricky. So there is a mouse model where you can conditionally knock out FMRP. It, unfortunately, is not a total knockout. So you still get a slightly leaky expression of FMRP. C.E.T.S, who have treated me very well over the years, have recently developed a rat model where we can knock out FMRP at any point in development in a Cre-dependent manner or a tamoxifen-dependent manner. And we have just, we've got a postdoc in the lab working just now on exactly that question. At the moment, what we see in the rats recapitulates some aspects of what's been repeated in the mouse, but we've never seen these big changes in spontaneous frequency that have been reported by others.
Okay, other questions? Yes.
This is more of a philosophical question, but we have some time, I guess. When you put the tissue in culture, the normal rhythms of changes in oxygen tension, changes in blood pressure, the sounds, the neurocircuitry, the ebb and flow of gammas and theta waves are out there. But I wonder if you can have maybe a more focused way of changing like the extracellular milieu locally using cage glutamate or cage GABA, because there's a steady stream of transsynaptic stuff, and using propofol studies. And the final question is there any effect of anesthesia like propofol on people with Fragile X disease, or is it unusual effect in terms of the expression of the transition to consciousness and recovery?
That's a really good question. I don't actually know about whether propofol has an effect on Fragile X individuals. Anecdotally, there's pretty well described in the rodent Fragile X field that rodents do not respond as well to isoflurane and volatile anesthetics as wild-type controls. So it's kind of one of these weird quirks. If you've got a Fragile X mouse versus a control mouse, you kind of have to get someone else to anesthetize them for you, because the knockout cells will, rats.
You're saying that the neurons are not necessarily replaceable modules, like going into the they're constantly changing their dynamics.
Yeah. So I'm not saying this is a perfect model, and certainly we don't have like wholesale circuit dynamics. However, what this does have over the other models, the extant models, is that we have recapitulated the diversity of cells that exist in the brain, and they have the human-specific genetic background that may account for some of the differences between what we see in rodents and what we see in humans.